Awesome. Thanks so much for having me. Excited to talk about this work. Yeah, this is the kind of stuff that I don't, as you heard about my sort of current job using more generative modeling and molecular data sets, I don't get to talk about this work quite as much. So I'm pretty excited to share this with you all and also to sort of catch up with old friends. Okay, so let's just go ahead and get started. As I think most of the folks here are familiar, we study sort of biological systems at a variety of scales, and because biological systems are very complex, we kind of tend to abstract depending on the scale that we're looking at, and everything from molecular work where you're kind of trying to study protein or molecule structures and properties and how they interact, to sort of signaling networks within cells and how they influence the sort of behavior of those cells. Individual cell models, so sort of trying to think about cells as a unit and their sort of like gene expression and how they might interact a little bit into groups of cells, and then last sort of like organisms, and then of course we have communities of organisms and so on and so forth. So today I'm going to be mostly talking about this sort of single cell and subcellular scale, but I've kind of been working across these scales throughout my career. In particular, I'm going to be talking about this idea of controlling cells externally using computational algorithms. This has sort of paralleled the advances in our ability to manipulate genetic material, whether via different molecular techniques to basically modify DNA within cells, and then trying to convince those cells to do things using either optogenetics, which is what I'm going to be talking about today, or sort of external chemical stimulants, et cetera, to kind of make cells do what you want. So that's the sort of control aspect. And then, of course, there's the quantification aspect. So when you're looking at cells, either under the microscope or through some other means of measurement, you have to kind of invert whatever you measure to what's actually going on in the cell. You very rarely get to observe directly what a cell is doing. And there's been a lot of cool work in this domain, in particularly with optogenetics, for the last 8 years or so. Particularly, so some of Jacob's work on hybrid digital systems, building hybrid oscillators in bacteria. People have used these methods to study feedback mechanisms. So you can imagine sort of mutating genes in a cell to delete particular feedbacks, which you think are important biologically, and then trying to restore those feedbacks via some synthetic optogenetic or other pathway, such as the sort of work I'm showing in the bottom left corner by Hanel Smad's group. You can imagine trying to drive transcription or gene expression to desired levels in individual cells, and that's going to be these studies on the right using the EL222 system by Mustafa Kamash's lab, and then by Jean-Baptiste doing some deep learning to build spatial patterns using sort of deep learning based control. So this is it's like a super exciting and cool field, and so we started working on this a couple of years ago. And let's just kind of go to the beginning. I know folks here are probably very comfortable with all of this, but in the sort of typical control view, you have the value that you want your thing you're trying to control to take. So the set points, in our case, that's going to be the number of molecules over time. And then you need a way to control each cell. So in our case, I'm going to be talking about a digital micromirror device, but you could use other things. The plant is just the cells. You need to observe them, and then you get an output, which may or may not be the same thing as what you're sort of controlling for, right? So you measure fluorescence, but you want to control, for example, the number of molecules in the cell. And in the sort of biological setting, it's not always clear like what your controller should be. You can use a data-driven controller, like some deep learning based thing. You can use model predictive control. You can use things like PID control, so on and so forth. And then how do you sort of take your measured object, in this case fluorescence, and use that to build a feedback in your system. So I wanted to say at the beginning of the talk, but if folks have questions, please interrupt me. This can be informal. Okay, so let's talk about those fluorescent maps. So how do we say like we want to control the number of molecules of a particular protein in a cell? So a cell has a given number of those fluorescent proteins. Each of those fluorescent proteins is going to emit a stochastic number of photons, and then you capture those photons in your camera. And so there are several layers of complexity and abstraction just in the emission and capturing and conversion to digital measurements which occur. And we're going to, in this work, kind of take some approximations around large numbers of those photons. So I'm showing at the bottom here the what happens when you assume that each protein in your cell is emitting a Gaussian distributed number of photons, which of course is only valid when you have really bright fluorophores. And so and then you convolve a bunch of those together, and one for each protein in the cell, and then you get some fluorescent measurement. So here P sub n is going to be the probability of having n molecules, n proteins in a given cell. And then you can see we just have the Gaussian expression on the right. So now the question becomes, how should we control cells? So there are kind of like different ways that you can imagine doing this. So there's population-based control, where every controller gets, every cell gets the same control under the microscope. So this would be if, it doesn't even need to be necessarily a microscope. It could be some kind of bioreactor system or another system where basically you're giving every cell the same signal. And then this is kind of contrasted with the single cell control, where you're trying to control each cell, give each cell its own input. And different modeling paradigms are kind of appropriate for these different scales or levels of control. So in the population control, we get to make use of population averaging. And then we can kind of use central limit theorem approximations to say that we have Gaussian dynamics. And we can use traditional sort of modeling or filtering and estimation like Kalman filters. However, when we have each cell gives its own controller, has its own controller, we might have non-Gaussian discrete dynamics of the underlying gene expression pathway. And so we need to use something that can work with those kinds of dynamics. And so we use a sort of more general Bayesian filtering approach. So let's illustrate the importance of the sort of like stochastic modeling for a simple toy model, and then we'll build towards more realistic systems throughout the talk. So I'm going to consider just a self-regulating gene, where it's you have some gene which has produced X, and that protein, a gene which produces a protein X, that protein X degrades, and the the protein X is sort of upregulating itself. So you have a Hill function that is saying if there's more X, then there's going to be more X. And then you also have this controllable element, which basically says I can move up and down the baseline production of X with some control U. So let's look at two different kind of views of this model. And so on the left, I'm showing two traces. Basically, the dark orange is going to be just a deterministic representation of the system. And you can see if you start, let's say 100 molecules, over time, you're going to decay to some steady state, and your system will just stay there. Meanwhile, the sort of light orange is a stochastic simulation of the same system, same parameters, same everything, but you can see some interesting behavior. While it mostly starts and kind of oscillates around that and fluctuates around that higher mode, that higher steady state, it occasionally will kick down to this lower stable equilibrium point. So if we're looking at the plot on the right, you can see that the dashed line, so the decay, crosses the Hill function. The solid black line at 3 points. The two outermost points are the stable equilibria of the system, and then in between is an unstable point. And you'll notice that in this parameter regime, the system kind of fluctuates around the high equilibria, but then it will bounce down past that unstable equilibria into that lower stable state. It'll stay there for a little bit and then bounce back. And so if you were trying to control the system using the sort of deterministic viewpoint, you're probably going to do something very different than if you are considering, because you would never observe this sort of like state switching if you're using that as your model. So you would never predict that you would leave this sort of equilibrium point. So we want to use a stochastic model when we're talking about working with single cells. And so we're going to turn to the chemical master equation, which describes the sort of time-varying statistics of these gene expression processes. So it's actually a very sort of easy to write down set of equations. So this is going to be P, so looking at the middle here, dp dt equals some generator matrix times P, when P is your vector of your probability states in your system. And when you solve these ODEs, you get something like I'm showing on the right, which is the time evolution of a probability distribution of your species of interest over time. Now, of course, this is computationally expensive and intractable when you have like a bunch of species in your system. Typically in these kinds of control models, we're just considering a couple of species. And so in principle, these might be tractable. But of course, you can have this sort of like computational nightmare with working directly with the chemical master equation. So we use the finite state projection approach to truncate the chemical master equation. And the sort of trick of this approach is that you pick the states which have high probabilities in a finite time. And every reaction which makes you leave your finite state space moves you into a sinc state, which we call G of t. And when you do that, you get instead of an infinite set of ODEs to solve, you get a finite set of ODEs. And you get this like additional ODE, which describes the flow of the probability from your states you kept into a sinc state. Now, of course, if you wait long enough, your system will eventually, all the probability will end up in G of t. But for finite times, you have an exact approximation of the chemical master equation. So let's go back to our self-regulated gene and see how we can, if we can control it using a finite state projection based approach. So we have our system here, and then we're going to just define a simple control which is on or off. So you can either drive the system up, so the upper gray line here, and that kind of moves the equilibrium point higher. But it removes the unstable equilibrium in the low state. And then you can also have the low control, which does the opposite. And then we're going to use Bayesian filtering with the finite state projection based method to actually use this control. So at each time, you have the option of either putting the control on or the control off. And what that amounts to in our sort of chemical master equation world is just solving the master equation with two different generator matrices, which I denoted here as A on for the generator matrix when the control is high, and then you have just a different generator matrix when the control is off. And what this gives us is a mapping from a probability vector at some time t-1 to some time t, depending on which control I use. Okay, and then I need to estimate the state of my system. And so what I'm going to do is I'm going to use my measurement model, which is this F sub RFP, which basically gives me the likelihood of observing a particular fluorescence value, which is the thing that I would measure under the microscope, given some number of molecules in a given cell. And then I use my FSP model to give me a prior, right? So my prior at time k is just the propagated estimated state from my prior at the previous time. And so the difference, this is exactly like a Kalman filter, except for instead of only needing to keep track of the mean and variance, which is all you need, right? For a Kalman filter, we have to keep track of the entire distribution. And the thing which maps those distributions from time to time is the finite state projection algorithm, which again, so for each cell, I have a entire probability distribution of numbers of molecules in that cell. And that's going to be changing in time according to the model dynamics and according to the measurement that I've received. So there's our state estimation using a Bayesian filter. And then the last thing is I need to use a cost function to basically perform some kind of predictive control here. So what we did is something simple, is to just exhaustively enumerate all the possible inputs in our prediction horizon that we pick, which is of course sort of like a hyperparameter, and optimize this cost function, which we use the average absolute deviation of the system to the target. And again, we're going to use sort of this receding horizon MPC. So you predict out, for example, like in this gray region, what the optimal control would be, and then you only end up using a shorter version of that. So the model can see further into the future, but you wouldn't use all of the designed light sequence for that horizon, and then you just repeat this throughout. So for our toy example, we do this with kind of trying to restore or remove the state switching that was observed in the stochastic system. And so you can see here the orange and purple solid lines, the darker lines are the deterministic behavior of the model, and then the lighter lines are actually just Gillespie simulations, the thing that we'd be trying to control. And you can see that both of them are observing, you see switching between the states. And so we're going to see if we can use a simple controller to remove that switching. And you can see that even though the system cannot exactly track the target, you can reduce dramatically the amount of state switching that you see and kind of restore the deterministic behavior in the system. So this is cool, and it kind of highlights that there might be some utility to working with the single cell controllers. And we want to see if this is like usable in the real world. So in order to do this in the real world is obviously a huge effort. And so you need some kind of wetware. So you need something biologically that, has an optogenetic system. So we worked with the EL222 system, and then we put the promoter of that EL222 molecule under on an M-scarlet fluorescent protein. And then we basically did a sort of pulse width modulation to drive up and down the gene expression by pulsing blue light on the cells. We then need some hardware. So we need to be able to send light to individual cells, and we need to keep the cells alive and happy. So we use a digital micromirror to send light to cells. We use microfluidics to keep the cells happy, and then we observe them with a microscope. And then the other sort of key ingredient is the software and algorithms. So I talked a little bit about the control algorithm, the sort of Bayesian filtering and that kind of thing. But the other key component is going to be the image analysis and photostimulation and controlling the digital micromirror and controlling the microfluidics and so on and so forth. And I'll talk a little bit more about the software that we developed to do that next. So here's how some cells growing and dividing, some yeast cells growing and dividing under the microscope look under the blue light. And you can see that you do get differential expression. There's definitely some stochasticity of these cells in their expression. Okay, so we need to model this sort of because we're doing, again, model predictive control. So we need a model for our system. So we basically modeled the EL222 production, and then the dimerization, and then finally the sort of like promoter dynamics of the M-scarlet protein. We have our microscope. So this is a picture of the lab at Pasteur, and all of the cool stuff that we used. We used a digital micromirror to stimulate each cell. So this will let us get sort of micron level precision of sending light. So in order to kind of like control different cells over time, we need to be able to track them over time, right? Each cell needs its identity, so we can know what its history of stimulation was. So we start with so we need to, in order to track, we need to segment. And so this was sort of before, in the middle, I would say, of all of the revolution of the cell segmentation algorithms that's now are sort of widespread and really easy to use thanks to like work of people like Jean-Baptiste. So we took the bright field images, we built some preliminary masks, and then I made a little Napari based mask adjustment user interface where I could draw the masks that were missing, and then retrain UNet based on delta, which again is from Jean-Baptiste, to segment yeast cells. And so you can see importantly that this like can work for both dense and for sparse fields of cells, and so we wanted to quantify sort of how well we can segment and track the population of cells as it grows and divides under the microscope. So the tracking is simple. We just used a TrackPy, so a standard sort of tracking Crocker-Greer algorithm. And so what happens here is like you have, the cells are dividing over time. So you need to detect them when they're created. And so you can see that occasionally we do not immediately detect new cells, but somewhat, and once they reach a sort of size that is reasonable, we do detect them, and then we can track them over time. So we basically did a whole lot of manual checking to see if we could correctly track cells. We found that most of the cells were correctly tracked for most of the frames. And when we do have errors, it was mostly due to either segmentation problems, or so those would be like an issue where you just miss a cell in a frame, or we would have cells flowing away, so sort of like experimental issues. We found that kind of when things were segmentation was working well, and when the cells didn't just sort of disappear or flow away mid-experiment, we could track the cells over time quite accurately. So all of this was put together with a ton of hard work by Stephen Fletcher, who I'm showing in the picture here, who's an engineer in the group at the time, into this software called Micrometer. And this is a sort of reactive microscopy platform, which lets you, it's written in Python, and it lets you kind of control your microscope and easily automate experiments. And there's since been, in around the same time, a number of similar software which come out that are aimed at sort of automating and building microscopy systems that can kind of take what they're looking at under the microscope and use them to change imaging conditions and so on and so forth. And so we did a number of other experiments with this platform besides the sort of control stuff that I'm talking about today, but you can read about that in our paper. Okay, so back to this. Oh, and another fun thing that I like to mention is that we built a little Discord bot, so you could kind of like query the microscope from home if you were so inclined. Okay, so the model. So we built this sort of stochastic model of the protein production. We have our measurement model, which is the same one I showed you all before. And now we're going to basically fit and validate the stochastic model of the sort of real system. And so here in red, I'm showing the measured single cell trajectories of some of these yeast under the microscope, which were experimented under the control shown here. So the dark blue means light was shining on the cells. The light, or the gray, means that there was no light on the cells. Here, the red lines again are the actual single cell fluorescence trajectories, and the green heat map is the inferred probabilistic model obtained by fitting parameters to those trajectories from our stochastic model, our FSP model. And so you can see basically that the darker green, the higher density should contain most of the trajectories, and indeed it does. We can track the mean very well. So if we just take the mean of our model of that probability distribution, it matches the mean of the data pretty well. It's not perfect. And then the same is true for the variance in the model. And then we validated with a simpler experiment, so maybe a less rich dynamics, but you can see that these parameters were not changed, and then you just leave the lights on and see what happens in the model, and you can see that the behavior is captured pretty well. So this, again, it's not like we're trying to build the sort of optimal model with lots of complexity to capture all of the biological behavior in the system. We want to build something that is representative of the system and contains the sort of necessary components to control individual cells. So another cool test of the model that we did is to perform just an open loop control. So I guess it's not control if it's open loop, but basically we performed an open loop experiment, which was designed using the model. So we have our we set a target, which was like this sort of step up based target for the molecules. We searched over the input space over that time horizon and came up with this input signal. So which should track that desired number of molecules, and then implemented it at the population level to track the target. And you can see something interesting. So we see that the population mean tracks our target quite closely. Meanwhile, each individual trajectory is all over the place. And you can see there are lots of offsets there. So the population level control is kind of doing what we would expect. It is creating a mean signal which matches to some degree the desired target, but the single cell variability is super high from cell to cell. So then we wanted to implement our closed loop control and at the single cell level. So and this is using the Bayesian filter that I described earlier. So we first validated this using Gillespie simulations where there's no model mismatch, right? So here, we know that what the simulated cells are doing is exactly what the model says they should do. Of course, there's still variability because you have a stochastic fluctuating trajectory. So we can see that it's tracking the target nicely. We can not only accurately estimate the number of proteins. So here the red line on the left is going to be the true number of protein in the simulated cell, and then the green heat map is the estimated number of the observed protein, the distribution of that. So you can see it's like very sharply peaked. Only a few pixels are very dark and the rest is light, which means that our estimate of the proteins are sort of high confidence. And then we can also estimate the hidden state. So basically the number of EL222, not the number of M scarlet proteins. And you see that it's much more blurred and there's higher uncertainty, but still is like somewhat generally close to the true number, which again is the sort of dark solid line shown through these plots. And then we did some further comparisons between the sort of population level controls and the single cell controls to understand when population controls are good, what are they good at, what are the sort of limits of the single cell control, what are the trade-offs. And so here on the left, I'm showing this is all still simulated. The simulated control for the population. And you kind of see what we saw in the experimental results, where the mean tracks nicely, but you have kind of a lot of variability across single cell trajectories. Whereas for the single cell control, you have a much more precise control. And so we quantified some of this. Oh, sorry, that's blurry. The population level control has a higher steady state error. So this was in an experiment, which I didn't show, but where it's just drive the cells to a certain number of molecules. And that's what we define as the steady state number of molecules. And you can see that the error is higher for the sort of population control than it is for the single cell control, regardless of the population size that you use, which is kind of an interesting observation because you might expect that as the population size gets larger, the difference from the mean to the steady state target level should go to 0. But because of the stochastic dynamics and the approximation induced by the linear noise approximation, you actually still have kind of a mismatch. And so you don't precisely follow the mean there. We also showed that, and you can kind of actually see it here, the time to target. So if we look back at this plot, if we look at just around 200 minutes, where the signal goes from the low state to the high state, you can see that the mean, so the darker red line of the population control on the left, kind of achieves the higher level, the higher target level faster than the single cell control. And we observe this across population sizes for the for these two approaches. So it's kind of interesting that the population control might be a little faster, even though it's less precise to achieve a particular target value. Okay, and then finally, we implemented the single cell control in real cells. And so here I'm going to just show a video from one of the experiments where we are, you're going to watch the cells kind of growing and dividing, and that's the sort of purple color, and then you'll see flashes of yellow. And those flashes of yellow indicate the signal being applied to those cells. And then we'll look at the results afterwards. So let's take a look. You can see each cell is getting kind of different signals. And then at some point, you'll see a big flash of all the cells, probably in a second here. Right now. And that's where the target went up. And then it's kind of just sort of, the algorithm is poking each cell to maintain that level. So we can look at the results and the controls for each cell here. So again, each individual trace is going to be a different cell. The population mean of all of those is dark red. And you can see that we can closely kind of track the target. And then we compare this to the population control, where every cell saw the same thing. And you can see that we've got much higher sort of variability. And you also see that the signal's going sort of like, much higher for the population control. Okay, so I'll wrap up now and take questions that folks might have. So you can see that, we've got amazing biotechnology that lets us do all of these cool manipulations and controls of cells, particularly using optogenetics. If you're going to talk about building model predictive controls for single cells, you want to think about the stochasticity and the non-Gaussian components of those models, because they do affect the sort of precision of your controller, the ability of your controller to achieve the target. And I think looking forward, there are lots of interesting things to do. For example, designing light patterns to optimally learn model parameters, to do that simultaneously while trying to control the population being observed, and perhaps doing sort of like fancier or more exotic targets rather than just controlling the gene expression of a single gene in a single cell. You can imagine more complex scenarios where cells are communicating and trying to build sort of like simulated cohorts of cells. So I think there's a lot of exciting work to be done in that direction. So with that, I'd like to thank Stephen, who was sort of the huge part of this research, particularly building the platform, the Micromator software, the sort of implementation of this into the wet lab. And then also with to Jacob and Gregory, who are here, who were my postdoc mentors. We did this work. And then also sort of to folks who have kind of like helped before and after at CSU and Los Alamos. And again, I'm now at Oak Ridge National Lab. I'm a research scientist there. And yeah, so if you're interested in talking more or collaborating, potentially taking this to another step, I would love to hear your thoughts. So feel free to shoot me an email. So with that, I can take any questions that folks might have.